Good morning, everyone. So our topic for today is the effects of tocotrienol on osteocyte lacuna network and osteocyte-specific bone peptides in osteoporotic rats induced by high carbohydrate, high-fat diet. So these are my group members, and these are my supervisors. So I'll start off with metabolic syndrome. So what is metabolic syndrome? It's a complex of interrelated risk factors for cardiovascular disease and diabetes. So the factors include central obesity, high fasting blood glucose, high blood pressure, high triglyceride, and high density lipoprotein cholesterol. So what is the relationship between metabolic syndrome and osteoporosis? So metabolic syndrome has these components: increase in abdominal circumference, decrease in HDL-C, increase in blood pressure, increase in triglyceride, and increase in plasma glucose. So all these components will lead to release of cytokine, inhibit osteogenic activity in vascular cells, hypercalciuria, and reduce blood flow to the bone. So overall, it will increase the osteoclast differentiation, decrease the osteoblast maturation, increase the inflammation, increase the calcium excretion, which will lead to increased risk of osteoporosis. So what is osteoporosis? It's a disease characterized by low bone mass, deterioration of bone tissue, disruption of bone microarchitecture. So this will lead to weaker bone and a higher risk of fractures in elderly. What is bone remodeling? It's a continuous process of synthesis and destruction of bone in mature structure. So this will governed by three components, which are osteoblasts, osteoclasts, and osteocyte. So if bone formation and bone resorption is not tightly regulated, osteoporosis can occur. The role of osteoblasts and osteoclasts is widely investigated and research has been done, but the role of osteocyte is not fully understood. So for role of osteocyte, it mainly secretes two peptides, which is DMP1 and PHEX, which will regulate phosphate metabolism. So PHEX will degrade PTN, which PTN is phosphatonin. So if PTN is not degraded, it will remain active and downregulate NPT2. If less PHEX is produced, more active PTN will be available, which will lead to reduce in phosphate absorption and increase in phosphate excretion. So DMP1, it will carry phosphate to extracellular matrix, providing phosphate for mineralization, which will in order for osteoblasts to mature to osteocyte. So there's a lot of studies have been done and carried out aimed mainly to develop an alternative osteoporosis agents using natural products. So one of the natural products that has been receiving a lot of attention is tocotrienol. So you might be asking me, what is tocotrienol? So tocotrienol, along with tocopherol, is a family, belongs to a vitamin E family. So the main source of tocotrienol are annatto bean and oil palm. So they have this property, antioxidant property, reduce expression of pro-inflammatory cytokine. They also downregulate mevalonate pathway and enhance the expression of bone formation gene. So overall, they will reduce the bone loss. But the effect of osteocyte-driven phosphate metabolism is not fully understood. So our study here is focusing on that. So what is our question here is, does tocotrienol affect the osteocyte lacuna network and osteocyte-specific bone peptides in osteoporotic rats induced by HCSF diet? So our objective is to investigate the effects of tocotrienol on osteocyte lacuna network and osteocyte-specific bone peptides in a rat model bone loss induced by ACSF diet. So our specific objectives are to measure the effects of tocotrienol on osteocyte number, empty lacuna number, DMP1, PHEX, and phosphate level in osteoporotic rats induced by ACSF diet. So our hypothesis is tocotrienol increases osteocyte number, decreases empty lacuna number, increases DMP1, PHEX, and increases phosphate level. So for methodology, the samples were taken from a previous study in which a total of 42 male Vista rats aged 12 weeks old were purchased, and they were randomly divided into 7 groups. So the baseline group was sacrificed after acclimatization. The normal group were fed with a normal standard retro and tap water. So the remaining 5 groups were fed with high carbohydrate, high fat diet to induce metabolic syndrome for 8 weeks. So after the 8 weeks of diet, the treatment with tocotrienol with different doses were given to 4 groups of HCSF rats, and the diet was continued for 12 weeks. The doses used in this study were 60 milligram per kilogram day and 100 milligram per kilogram daily for each tocotrienol, another tocotrienol, and palm tocotrienol. So for this study, bones, femurs, and tibias were harvested. Left femur were subjected to bone histomorphometry, and left tibia and serum were used for measurement of osteocyte-specific biomarkers. This is our previous study. The chart shows how the process is done. So in the study, tocotrienol is stated It can act as anti-hypertension, anti-hyperglycemia, hypercholesterol, and anti-osteoporosis agent.
Next is quantification of osteocyte number and empty lacuna. We took the sample from the last femur, seen H&E, and we used the Weber grade to count the osteocyte and lacuna. Next, for the protein extraction and quantification, we use the last tibia, extract using RIPA buffer, and we quantify using Bradford method. We assess the bone protein and phosphate levels using ELISA kit, and for statistical analysis, our data is normally distributed. Therefore, we use ANOVA with Tukey and or Dinet's post hoc test. It is considered statistically significant when P is less than 0.05. Our data are presented as the mean and standard error of the mean. Moving on to our results. The first objective is to measure the effect of tocotrienol on osteocyte number and empty lacuna in the osteoporotic rats induced by HCHF diet. In this study, we found that the HCHF rats had significantly lower osteocyte number, as you can see in figure 1.1, compared to the normal rats. HCHF rats had significantly higher empty lacuna number, as you can see in figure 1.2, compared to normal rats. We also found that treatment with 100 mg per kg another tocotrienol was able to significantly reduce the empty lacuna in the HCHF animals compared to the vehicle-treated HCHF animals, as you can see in C. Okay, the reduction in osteocyte number and increase in empty lacuna in HCHF rats indicate an increase of osteoblast apoptosis leading to bone loss. A previous study showed that there were decrease in osteocyte number and no change in empty lacuna in avarexamized rats. Also, treatment with self-emulsified another tocotrienol did not show any changes in osteocyte number and empty lacuna. The slight difference in the findings obtained from previous study compared to ours could be due to the difference in osteoporotic model used and the formulation of the tocotrienol treatment. All right, this is the representative figures from the H&E staining. Arrowhead indicates empty lacuna and yellow indicates osteocyte. As you can see, there is more osteocyte present in the HCHF 100 and plus 100 another tocotrienol and more empty lacuna present in the HCHF slide. Next, our next objective is to see the effect of tocotrienol on the MP1 and PHEX, right? So our result shows that both the MP1 levels and PHEX are significantly reduced in HCHF rats compared to the normal rats. Previous studies suggest that in the MP1 now mice, morphological changes in bone and bone mineral loss are obvious. Lastly, the effect of tocotrienol on phosphate level. In this study, we found that there were no significant changes. We postulate that longer time is needed to observe the effect, as previous study by Chin and Imanyawana stated that the beneficial effect of tocotrienol on bone calcium take longer time to manifest. So in conclusion, the skeletal deterioration in the HCHF diet by animals is mediated through the reduction of osteocyte quantity and the level of osteocyte-specific bone peptides, suggesting that osteocytes are important coordinators for bone metabolism. Treatment with tocotrienol for 12 weeks are insufficient to reverse the changes in these parameters. How our research contributes to the current knowledge is that this is the first study that helps to investigate the role of osteocyte in regulating phosphate metabolism in osteoporotic rats induced by HCHF diet. The importance of our research project is that we need to understand how tocotrienol supplementation can be benefit to our health and therefore better life for the elderly and osteoporotic population. The practical application and potential impact of the research project is to educate people about the benefits of tocotrienol in treating a common disease, to encourage more research on tocotrienol, and to make full use of natural resources, which is the annatto seed and palm oil, to produce a product which can help with the economy. The limitation of our research is that the measurement of bone peptides was only performed at the end of the study. We were not able to monitor the sequential changes in their level throughout the study period. And also, osteocyte count at the trabecular region and the levels of the MP1 PHEX and phosphate in serum were not measured. Future direction, longer duration of tocotrienol should be provided to validate the results of the current study. Effects of tocotrienol treatment on other osteocyte-specific bone peptides should be investigated. This is the acknowledgement. These are our references. And lastly, that is all from us. Thank you.